if you have a model that you can trust, that is, if I think my model really represents the specificity of the protein, it tells me where I would find binding if I was just binding to naked DNA. Well, of course, in vivo, it's not naked DNA. It's all other kinds of things that are going on. So whenever I look at the discrepancies, it's actually telling me something about what's happening in vivo. When there are discrepancies between your predicted and your observed data, that actually tells you something of biological interest. If you have false positives where you would not predict there would be a binding site, but there is a binding site, it tells you there are interactions going on that you should follow up on. And then finally, we can use all this extensive data for improved recognition models, which will be very useful for transcription factor design. High throughput data can be very useful for protein DNA modeling. It's best when the analysis method is aware of or adjusted for the kind of data that's used.
You can see when we are comparing ourselves to a gold standard of Amadeus is the most accurate. The first part talked about motif discovery. I told you about the Amadeus platform. Its basic goal is to analyze the target gene set, but
more than half of these unverified transcripts may encode products that are non-functional in the protein sense. So long non-coding can do a number of things, including code, of course, for microRNAs, but they can also act as decoys to keep the microRNA from reaching their target. It's a new regulatory level, which we had no idea about. That means that you have genes, they are regulated by transcription factors. Transcription factors can be regulated by their own activation, but also controlled by microRNA. But now microRNA can be controlled by long non-coding RNAs, which can keep them from acting and therefore leading to the expression of genes.
Most of the long non-coding RNAs that we have in our data sets are relatively poorly conserved at the level of sequences. So sequence conservation is, as a group, above average. There is a significant signal, statistically significant signal for sequence conservation. So for selection pressure to maintain sequence in this region, you can find transcripts by just looking at the conservation of splice sites. And these non-coding RNAs are evolutionarily fairly old. We actually have about 1,500 that go back to the marsupials, where the alignments already get really bad. On the other hand, these numbers also strongly indicate that we have a lot of turnover or a lot of differences between the conservation of individual splice site, the structure of these genes in individual mammals.
On one side, the community wants the reference genome to be complete and accurate as possible, and on the other side, it doesn't want it to change. And this is kind of a fundamental problem. We're keeping the reference as it is, but we're releasing these patches. So this is the future. Most patches will be novel. Many of them will have important functional consequences. We already have a GWAS hit, which is totally uninterpretable until you switch to the patch version, where suddenly you have a signal. This has consequences for you lot, because it means all your mapping algorithms need to become patch aware.
And all the interesting stuff, quite likely, is in the blue regions. The intron regions, the intergenic regions, the regions that don't end up in protein. Because those are the regions where there's potential complexity that is able to pick and choose when these protein coding regions should be put together, rearrange those protein coding regions to do different things. So that's what I think about as the regulatory code, the sequences sitting in these intergenic regions. What do I mean by regulatory code? Well, it's something that extracts features from DNA sequence, and then depending on cell type, can predict the mRNA level.
So the question for today's talk is, who's GC? Conclusion, GC bias is not simply determined by the ratio of the GC count to the fragment length. I've suggested that if you're going to have one region of GC which appears to influence things landing on the genome, it is the window which is the fragment length minus the ends. This is the summary. We seem to have ruled out GC content of the read, which is the most obvious one. We seem to have read GC content on a scale more global than the fragment. Base composition around the fragment breakpoints is a noticeable role, but doesn't explain stuff. Speculation over causes is left for another day because lots of stuff is going on. There seems little doubt that PCR amplification of the fragment accounts for the majority.
In junk DNA, we could actually encode secret messages, and so maybe there was an alien civilization somewhere that has encoded secret messages in junk DNA. We talked about first the non-coding functional annotation. We talked about the outputs, the non-coding RNAs, and the big message here, I think, was the data integration to identify non-coding RNAs, the fact that some of them were pseudogenes, and those pseudogenes potentially had a regulatory role. The TF network connected to the microRNA network, and it created these kind of interesting motifs. Also, how you could kind of create a kind of hierarchy, and how you could rationalize some of the phenotypes here. You know, for instance, the essential genes and whatnot, in terms of the information flow bottlenecks in this hierarchy. A lot of these non-coding sites, such as TF binding sites and non-coding RNAs, are under selection. And they're under a different type of selection, particularly when you look at the full spectrum of variants such as indels and SVs, than the type of selection you see for protein coding genes in terms of their sensitivity for indels.